Hello everyone, welcome to the online classes. So in the previous class we had discussed some of the introductory parts of edible vaccines. We all know that edible vaccines are the vaccines which are used or we basically consume it in the raw form. So taking that into consideration, so we have some of the examples. One what you can say is banana, the next one what you have here is the watermelon. So now what is the procedure? In my previous class I discussed the general procedure for production of these edible vaccines. So today we will discuss the exact steps which are involved in production of these edible vaccines. Now if you try to consider here steps involved in edible vaccine production, the first and foremost thing is embryogenic cells. Now what is this embryogenic cells? We try to take out the plant cell, something like an egg plant. Make sure that this egg plant gets converted to its embryonic stage. So that is one of the key factors to produce a edible vaccine. So what we try to do here is we try to add some of the growth factors. As we discussed in the previous classes, cytokinin auxin with a proper ratio, we do get embryogenic cells, something like a somatic embryo. So here we are trying to produce a somatic embryo by adding either increasing or decreasing the ratio of growth factors, cytokinin as well as auxin. So from egg plant, once we have got this embryogenic cell, the next what we try to do is infection by bacteria. Now infection by bacteria, we would try to already construct the vector system here. It may be either binary vector or it may be either co-integrated vector. So gene of my interest is being incorporated into the vector system, and then that bacteria is allowed to infect this cell. So once infection is done by this bacteria, now within the plates we have bacterial cell, we have the embryogenic cells. I have to differentiate between bacteria as well as embryogenic cells. So what we try to do is we go for an antibiotic treatment. Once we do this antibiotic treatment, the bacteria would be killed. We don't want bacteria now, we wanted only the cells. So my interest here from the third step is I wanted to kill the bacteria. Once bacteria are killed, next thing is I'll get only the egg plant, or I'll be getting the cells basically. I'll try to collect these cells, go for culturing. Once I culture, I'll be getting a regeneration, or I'll be getting a, take an example, a complete plant. This is considered to be as regeneration. Once I've got this regenerated plant, it may be either the fruit, leaf, stem, root, irrespective, whatever you are trying to eat in the edible or in the raw form, we'll take those components, either fruit, leaf, stem or roots. We'll go for extraction of the content and we'll go for quantification. Now this extraction and quantification is one of the key factor to determine the percentage of antigen present. So once we come to the percentage of antigen by SDS page, we try to check what exactly is the percentage of antigen produced by any of these things, we go for distribution. So these 6 stages or 6 steps are key factor in producing edible vaccines. Now let us go to the example part. The first example what we have is banana and hepatitis B. Now if you try to see, I have mentioned 2 important things here, why and what. Now why banana? That is the question. Banana is one of the chief what you call it as vegetable, sorry not vegetable, it is a fruit what we try to get worldwide. No parts of banana is consumed in the cooked form, so it is always consumed in its raw form. So easy availability worldwide, it's quite cheap, and what we eat is in the raw form. So these are some of the key factors for choosing banana, and hence we use this banana as edible vaccine. Now going to the next question, it's what? What is hepatitis B? Hepatitis B is a kind of disorder or disease which is caused by hepatitis B virus. Now what basically this virus try to do? It enters into the individual. They will try to act upon this hepatocytes, that is the hepatic cells, liver cells. They will try to damage, hepatitis B will try to damage the liver cells. In turn liver cell would be broken down as the organism keeps on multiplying. In this regards, it is found that around 2 billion worldwide are killed every year. Irrespective, we have this vaccine for hepatitis B. Even then, the death rate of hepatitis B is too high. So that's the reason. What is hepatitis B? It is the virus which tries to damage or degrade the liver cells. Why banana? Availability, raw form, and it can be accepted by different worlds. That is World 1, World 2, World 3. Now what is World 1, World 2, World 3 is? They are the hierarchies of the developed countries, developing countries and underdeveloped countries. If you try to check here. So how exactly I try to incorporate gene of my interest? It's by the process of agrobacterium. So we all know what exactly is agrobacterium, a biological mode of gene transfer technique. We have co-integrated vector, we have binary system. So these two systems, they try to help in incorporating gene of my interest into the plant cell. So what we try to do, I've taken a tDNA. In this particular tDNA, I've tried to retain left border and right border. I've tried to remove all the oncogenes. Once oncogenes are removed, I'll try to incorporate gene of my interest or the segments which I wanted. The segments what I incorporate here is NP2, EFPS, HBS, AG and NOS. Now what exactly are these things? The first one, it is neomycin phosphotransferase. So neomycin phosphotransferase is a chief segment which helps in replication process of the gene. Okay, and it also helps in giving or determining between transformed and non-transformed cells. Next what you have is EFP, that is ethylene forming promoter. EFP is a strong promoter which can drive the next segment, which is called as hepatitis B surface antigen gene. So this is one of the promoter, a strong promoter which can help transcribing this segment very fast, or it is something like a strong, promoter is a strong promoter. The next one what we have is hepatitis B surface antigen. So this is the gene which play or which gives the antigenic property for individuals once it enters into the human beings. The next one what we have is NOS, which acts as terminator sequence, and extreme left, extreme right you have the conserved sequence, left border and right border. Now what we try to do here is, let me try to explain in the form of a story. I have this segment from LB to RB. So this segment is incorporated into what you call it as either binary vector or co-integrated vector. So once it is integrated, what we try to do is develop embryonic cells. Banana plantlet cells can be converted, it has been converted into embryogenic cells. Now take this segment in the form of bacteria, inside the bacteria, allow them to infect. Once it infects, I'll try to kill the bacteria. I'll go for explant culturing. I'll be getting a complete plantlet. And this plantlet, whatever I'm getting, the fruit, basically the banana, tries to show the property of antigenic. Once it is incorporated into human beings, we produce antibodies against this. And what is the chief activity of HBS antigen is, it is used by the bacteria for attachment process. Now what we try to do here is, we try to produce an antibody so that the attachment process itself is stopped. Though the virus enters into my body, if there is no attachment to hepatic cells, there is no breakdown or there is no degradation of hepatic cells, and hence though the entry of hepatitis B is observed, it cannot replicate. So that is the first example. The second example what we have is watermelon as well as measles. Again the question lies, why watermelon? Edible, raw form, it has a thick protective layer, so we need not worry about the storage process, and it can be transported anywhere. And what exactly is this measles? It is a kind of disease which is caused again by a virus. So it is a paramoxy virus. So what is this paramoxy virus, or what exactly this measles mean? Measles is a kind of respiratory disorder this respiratory disorder, once the organisms enter into the respiratory tract, we have severe symptoms, or we have some of the symptoms like headache, vomiting, we have fever, body aches. So with the help of blood streams, the organisms will move from one part to other part, from respiratory tract to maybe digestive system, from there maybe something like excretory system. So they will try to multiply, they will try to damage each one of these organs. So that is the seriousness of this. Around 10 lakh every year people are killed because of this disease. So that's the seriousness of measles. If you try to check here, again the sequences, the process is almost one and the same. What exactly is the mode? It is agrobacterial mode of gene transfer. Now what are the genes which we are trying to extract? We are trying to take two kind of genes here, HNG genes as well as FG genes. So what is the role of HNG gene is hemagglutinin muramide glycoprotein. So what exactly this HNG protein or products do is they help in viral release factor. Once maturation of these viruses are done within the cells, they have to be released. So in order to release, they wanted the activity of this gene. My interest here is let the organisms multiply within the cell, and I will make sure that they are not released from the cells. It makes very simple. Multiplication is observed, one virus has entered inside the cell, multiplication has happened, but they are not released. They would be precised or they would be stagnant to one particular cell. Later on our body cells or our immunity will try to break down those cells. So that is one mode. The other mode what we have is we have another protein called as FG protein or FG gene. FG gene helps in production of fusogen or fusion glycoprotein. Now what is the role of this fusion glycoprotein is they help in viral attachment onto a particular surface. How HBsAg acts? Same way FG gene also has an attachment process. Now initially the virus is not attached, they don't get entry. So that is the role of fusion glycoprotein. By chance FG gene is inactivated, virus has entered inside the organism or inside the cell, they have to be released outside. This will make sure that the virus will not come out of the cell. So these are the two important processes of producing, or these are the two important examples of edible vaccines what is being observed even today. Thank you for watching the video.